Derbicea is a type of hair algae often found in marine ecosystems. It is also a primary producer that photosynthesizes to provide energy to other species of animals. Chemicals like phosphate play a strong part in the optimum conditions in which this organism can tolerate. We decided to choose phosphate as our limiting factor because it is a common chemical found in runoff, which leads to eutrophication, which can in turn affect the growth rate of algae. This experiment was conducted under the hypothesis that there is a positive correlation between phosphate levels in water and algal growth. To conduct our experiment, we collected approximately 6 grams of Derbicea or hair algae from the marine aquarium in front of McKinney at Bridgewater College. After the collection of algae was sorted, a wet mass was measured and then distributed among the replicate samples. The solution that the Derbicea samples were placed into was a combination of marine concentrate, deionized water, and a designated concentration of phosphate in the form of monopotassium phosphate. The varying concentration of phosphate in each experimental group simulated a small-scale eutrophication event in which the effects of phosphate level on algal bloom growth could be determined. The experiment consisted of 3 experimental groups, each containing 10 replicate beakers, with each experimental group containing a different concentration of phosphate, those being low, medium, and high. After the experimental setup, 0.2 grams of Derbicea algae was massed and then placed into each individual beaker. The replicate beakers were then placed under a cool fluorescent light and left unsealed to promote growth over a 10-day period. Throughout the 10-day experimental period, observations of the algal growth were made every other day. On day 8 of the experiment, the algae was observed to be white in color with condensed filamentous structures which appeared to be premature death. The dead algae from each experimental group was then harvested, dried in an oven at 100 degrees Celsius, and a dry mass was then calculated. The average dry mass of the low, medium, and high phosphate experimental groups showed that the medium phosphate condition had the greatest amount of growth. The high phosphate experimental group experienced slight growth, and the low phosphate experimental group experienced little to no growth. These findings do not support our original hypothesis, which stated that an increase in the absorbance of phosphate correlates positively to an increase in the algal rate of growth. Therefore, our hypothesis is rejected. While we did not receive the expected results, the results of our experiment could potentially be applied to the concept of eutrophication-induced algal blooms in aquatic ecosystems. Our data may suggest that a minor eutrophication event where phosphate levels are only slightly elevated will produce greater algal bloom growth as compared to a major eutrophication event when levels of phosphate are dramatically increased. This could be beneficial to the prevention of algal bloom growth by controlling runoff and other environmental processes that lead to eutrophication and expansive algal growth.